Hi, I'm Rhys Coleman, Waterways and Wetlands Research Manager in the Applied Research Team at Melbourne Water. Thanks for joining us for another edition of Melbourne Streams, where we're showcasing research coming out of the Waterways and Wetlands Research Program. In this first series, we're releasing a talk a day based on topics voted on by the Waterways and Land Crews. Today's talk is from Dr. Anna Miranda of the Aquatic Pollution Prevention Partnership, or A3P, between Melbourne Water and RMIT University. Anna is an ecotoxicologist and has a keen interest in understanding the impacts of pollution on aquatic organisms, from algae all the way through to marine mammals. Anna's talk today is an introduction to ecotoxicology and what it means for the management of waterways. Anna will talk about current approaches to ecotoxicology, but also where things could evolve into the future. There's also links between Anna's talk and the research that we're undertaking in A3P, such as Sarah Long's MERI project, or Monitoring, Evaluation, Reporting and Improvement project, that helps underpin tracking of our Healthy Waterways strategy. If you have any technical questions about this talk, then please contact Anna directly. Her contact details are in this presentation. If you have broader questions about the Waterways and Wetlands Research Program or A3P, then please contact myself or Slobodanka Stojkovic, who's also in the Waterways and Wetlands Research team. Of course, now you can search Research Hub on the intranet. You can browse the various research projects that we've been doing in the past 10 years or so and get access to various reports, papers, presentations, and so on coming out of that research. I really hope you enjoy Anna's presentation today, and we look forward to providing more talks in the days to come. Thank you.
Everyone, my name is Anna and I'm a researcher at the AQUEST group at RMIT University. I'm going to introduce you to ecotoxicology, the importance of this discipline to manage the health of our waterways now and how we plan to use it in the future. Our waters support a wide range of species across different taxa, including species that we see when we walk through a creek, such as frogs or fish, but also invertebrates, algae, and other tiny creatures that we cannot see unless we use a microscope. All of these species are connected and are essential to the equilibrium of the ecosystem. However, the water quality is declining. People always settle where the water and the land meet. Large population centers are near rivers, lakes, coastlines, and with the increase in population comes the demand for chemical production, for personal care, for pharmaceutics, but also for agriculture and industry. Chemical monitoring programs have made a huge progress in determining a large number of compounds in the water. A large number of chemicals have been detected in our waters, from the well-known heavy metals such as copper or zinc to insecticides that often run off from our backyards, but also newer chemicals such as those that we use for our own personal care, such as antimicrobials that come from our soaps, UV filters, or even fragrances from our favorite perfumes. This data, however, won't tell us how healthy the waterway is. It will tell us there are pollutants here, but what does it mean to the aquatic organisms that are exposed to these compounds? To determine what it does mean, we have to assess the responses. And for that, we use ecotoxicology. This discipline combines the knowledge of other fields, such as ecology, toxicology, environmental chemistry, and ecology, and evaluates the effects of pollutants at different levels that range from molecular up to the ecosystem level. Quite often, however, those responses are measured at the individual level. Organisms are exposed to certain concentrations of a pollutant and their responses are measured. There's two main types of tests that we use to measure those responses. There are acute tests, where organisms are exposed usually for a short period of time to relatively high concentrations of a pollutant, and this type of test, that is the endpoint. After this short period of time, usually no more than 4 days, we assess how many organisms have died. And this will allow us to calculate something that we call LC50, which stands for the lethal concentration that kills 50% of the tested organisms. And these values are usually needed for further setup of guidelines. This type of toxicity is still widely used and is the first step to determine the effects of a pollutant, and is particularly important if the toxicity of the compound is unknown. So this is the way to start. Of course, there are other types of tests, and usually we do it after we have an idea how toxic the compound is, and we call them chronic tests because they usually run for much longer periods of time. Usually, organisms are exposed to much lower concentrations of the pollutant, and our aim is to determine which is the concentration that still makes an effect or produces a response. And those responses usually responses that are very sensitive to lower concentrations of pollutants, such as reproduction, growth, or other physiological responses. So to decide if organisms are likely to be impacted or not in the environment by a certain chemical, we often use those individual responses, like I mentioned, for example, the LC50, to make a probability distribution that we call species sensitivity distribution. For this, different species, as they have different responses in terms of sensitivity, which means here, for example, for cadmium, Daphnia magna, which is a little water flea, they are affected at lower concentrations than, for example, Daphnia carinata, which is the Australian water flea. The aim of this distribution is to allow us to calculate or to extrapolate a hazard level using a specific percentile of the distribution, and usually we determine the hazard concentration for 5% of the species, which is the HC5, which is the level that protects 95% of the species. On top of it, we will then divide this value by an extra safety factor to determine a predicted concentration of no effect, which is a value that we will use in the environment to protect a whole set of species. Often those responses that we use to determine the predicted concentration of no effect are obtained using a standard group of organisms like you see in the picture, and they're usually from the northern hemisphere, which can be very different from the ecosystem that we intend to protect. And it's because it's impossible to test a whole range of species, and most of the reference values for toxicity come also from the northern hemisphere. In the future, we hope that ecotox will be a more integrative science, that will see a move towards a more field-based approach instead of using laboratory-based tests where you use pickers to mimic the environment. The use of microcosms, for example, which are containers that contain sediment in a layer of water, which mimic the ecosystem, will allow us to evaluate those responses using local species, as well as having in account inherent environmental factors. Moreover, those molecular approaches such as DNA metabarcoding could open up a whole range of possibilities by quickly identifying a whole range of species that can also be a target for pollutants and would otherwise be impossible to assess, such as the microbial community. The DNA metabarcoding is a technique that allows us to determine which species exist in a sample by the presence of its DNA. This method of species identification will use a short sequence of DNA of a specific gene or genes as a probe. Then, after amplification, the resulting sequences are compared to a reference library, where each individual sequence can be used to uniquely identify an organism to species, in the same way that in a supermarket, a scanner uses those familiar black stripes of the barcode to identify an item in stock against the reference database. At the AQUEST, we've been following up on this idea, mainly looking at differences in microbial communities between contaminated sites and not so contaminated. So we selected two different types of environment, high heavy metal and hydrocarbon contaminated sites, and low metal and hydrocarbon contaminated sites. And we used sediments. From those sediments, we extracted DNA and we evaluated differences in the microbial community. And if we realize that those species that are specific, abundant, or they were more abundant in clean sites, such those here that appeared in this phylum here in red, as well, there were some phyla of bacteria that are characteristic in contaminated sites. This is how we see it in the future, and while we're still mainly using individual responses and then predict their responses at higher levels, we hope that we can directly assess those responses of entire populations, communities, and the ecosystem function itself. These methods will continue to be developed as part of the A3P projects. One of them is the MERI, stands for Monitoring, Evaluation Results and Improvement, which is led by Sarah Long. If you like any information of the broader context of the project, Sarah is the person to contact. But overall, what does it all mean for the waterways? It means that we will use a lot less surrogates, we will use more local species. Additionally, we'll predict less and we'll measure a lot more. All of this will allow us to deliver more accurate diagnosis of pollution issues, as well as more targeted and tailored management actions. If you think of anything that you might like to ask or share with us, you can get in touch with the team by sending an email or giving us a call.